is graph theory, on which these network techniques are based on, what actually the graph theory, and what are the node and edges. When we talk about networks, these are the common terms you will find in every network where we talk about node and edges, and what are the different types of network, different types of graphs. What are the steps in network pharmacology? If any one of you would like to apply network pharmacological techniques in your research area, so in what kind of research projects you can do by using the network pharmacological techniques. And then hands-on sessions will do on today's on first on how to identify the targets which are involved in particular disease. And disease can be based from your research area. So that hands-on session you will do on today's and disease can be based from your research area. Someone of you might be working on diabetes, someone of you might be working on neurological disease, and so and so on. So it depends upon your research area. So that hands-on session you will do how to create the disease network. So in network there is another term which is attached, is pharmacology. So pharmacology, when we talk about, we talk about study of drugs. But before studying the process, first we have to understand the disease, because unless and until if we don't identify the targets for the drugs, so that's why first we understand about the disease pathogenesis, or we are creating the diseased hello, screen is screen is Nivan, is visible. Weather is not okay due to that. Let me share it again. Is my voice is audible? Otherwise, I have to be connected with other connections. Let me not audible. Same. I'm going to join with if it's not visible. Just give me box. I have now connected with another device. Screen is not clear. Okay, let me share it again. But voice is okay. Mm, I think in every time it happens when you have to do the session, that time you will face the network issue. Okay, Gaurav is saying your voice is clear but screen is not visible. Yes, voice is okay now. Okay, now is screen is okay? Can anyone confirm it? Gaurav, Isa, now it's clear. Okay, sorry for the inconvenience, but every time it happens. Means whenever you start session or anything, that time you will face significant network issues. Otherwise, it's okay. Okay, great. So what I am telling you, there are 2 days workshop on network. You all knows this is a plan for 2 days. So it's a network pharmacology for the beginners. So if you are very new to this field and you heard about this term network, now every uh, one is talking about network pharmacology, but these techniques are not too much new. It's old terms, but only means name has been changed. Earlier we called it graph, or system biology. You might heard about system biology. So what these techniques are, so from starting from the day one, from today, what we are going to cover is what are the basics of system biology, what are the network pharmacology, why these techniques are important, where you can apply these techniques. That is the major objective of the workshop. The objective is, uh, if you want to apply these techniques in your research area, uh, I can understand every one of you have a different research area. Someone is working in the metabolic disorder, someone of you is working on neurology, in other areas. So whatever the areas, how you can in your research projects, how you can apply these techniques to enrich your research. So that is a major objective of this workshop. So as I said, you can see on the screens what we are going to cover today. So network is based on we are going to understand. Then what are the different types of network, what are the different types of graphs, what are the steps which you have to undertake if you are planning for a network pharmacological techniques. So first steps, you are going to do today, and you simultaneously you apply these steps and create some disease network. So what I am saying is, network pharmacology, when we talk about, you every one of you know that pharmacology means study about the drugs. But before the study about the drugs, you should know, or you should understand about pathogenesis of the disease, or the targets which are involved in that particular disease. So what are the different targets which you can hit by your drug molecules? So to identify the targets, first you have to understand about disease network. So today's we are going to cover up to disease network. Tomorrow we are going to cover how disease again lagging too much. Okay, okay, let me see how to deal with this. Now my voice is okay. Can anyone confirm it? Okay, great. So sir, what I am saying is, first you have to be understands about what is the disease pathogenesis. So before that, before network pharmacology, you should have a basic idea about what is system biology. So as I said, in system biology we are not looking at one gene, one protein, one pathway. No, we are analyzing how all these components, how all genes, proteins which are involved, and how they are interacting with each others and producing particular phenotype. So that is a system biology where we are not studying only at one gene or one proteins, but we are studying these all together. That is a system biology. And network biology is the one of the discipline of system biology, right? And network pharmacology is the subset of network biology. Are you getting my point? So system biology, as I said, system biology is not, we are not talking about single genes, single proteins. No, we are combining all genomic informations, transcriptomics, proteomics, metabolomics, epigenomics. So all together when we are studying, that is we call system biology. So at gene levels we call it genomics. When you, every one of you know, then DNA to RNA, RNA to proteins, then there is a process of transcriptomics that we call transcriptomics. When we are studying at the protein level, we call it proteomics. When we are studying at metabolite level, we call it metabolomics. When we are talking about how environmental factors are interacting with the genes, that will be called epigenomics. So all omics. So you can see that the last word is omics. Omics. So that's why you might heard about nowadays these word omic technologies, transcriptomics data, proteomics data, metabolomic data, epigenomic data. So we call it a big data analytics. ICMR is also conducting workshop nowadays, time to time on big data analytics. So what is big data? Big data is this one. Whatever the genomic data, transcriptomics, and to handle these data, this is very big data. To handle that, you should have a high performance computational systems. You should have a computer servers which have a large capacity to store your data, because there is a large data. So that's why we call it big data analytics. Thirsty, you might heard about, many of translational health institute at Faridabad, that are also dealing with the big data analytics. So what I mean to say, before get into the network, you should understand about its discipline as well. What is system system biologies, what it combines, what is the omic technologies, when we talk about big data analytics, what it is, right? So all these, that are the part of system biology. So if you ask me, what skills are required particularly, so if you ask me what skills are required to become a experts in network pharmacological techniques or network biological techniques, so I would say that what skill is required particularly. So skill, you should have a computational skill. Of course, you are dealing with the computers, you are dealing with the means most of the datas you have to be handle all the datas with the help of computer. So you should have a computational skills. But computational skill alone is not enough to become an expert in the field. You should have a very good understandings about the biology. So if you ask me, So going on for okay, if it's working, so that's fine. So so as I what I am explaining to you is, if you ask me what skill is required to become a good network scientist or network pharmacology scientist, the skill which is required is you should have a computational skill, you should have a good knowledge about biology, you should be you are very eager to learn new technology, because year by years new technology is coming up. You all knows this is the era of AI and machine learning, so all these techniques you have to be learn. You have to update yourself. Today's I am telling you some databases, tomorrow maybe there are new databases, so you can't say that no, I know only 5 databases, so that is enough. No, that is not the case. Maybe that databases has been updated and their name has been changed, so you have to be update yourself with new technology as well. So that is the circle which is required to become a good network scientist. So coming up with the, I can understand that many of or most of you are from the biology background, so you don't get, you don't want to get into the mathematics, but of course there is a mathematics is involved. Whatever the computational work you are doing, behind that there are a lot of mathematical equations who are working. When we talk about so this is a simple examples how mathematics helps in the predictions, because by the computationals what we are doing, we are predicting, we are creating the model, and that models are predicting the future. That model is predicting the possible mechanism of drugs in that particular disease, right? If we talk about network pharmacology, in network pharmacology what you are doing, you are predicting possible mechanism of xyz molecule. Molecule can be herbal, molecule can be marine compound, molecule can be new chemical entity which you have synthesized in your lab, and you want to understand what are the possible mechanism by which this molecule can play a significant role in that particular disease, right? By applying the network pharmacology. So what you are doing, you are predicting the future, you are predicting something, you are, how you are predicting, by creating the model. And what is the basis of models? To create the models you have to be do some mathematics, but of course there is no need to worry, because computer is doing all mathematical calculations, everything, but you should know about what is the process behind it. mm, Screen buffering, okay, screen is not, I think slide is still loading. Isa Garg is saying it's loading. Okay. Now, okay. Now I hope it's audible. Yeah, Jai Pharmacy is saying you are under poor network connections. Yeah, Jai Pharmacy actually, due to weather, there is a little bit, weather is not okay. So due to that, maybe there is a connection problem. As I said, whenever we have to do some online, always I face such kind of difficulties. Rest of times, network connection is too strong. But let me confirm now, is it okay, Isagar? Okay. So what I am explaining to all of you is, when we are getting the model is predicting the future, and model we are doing simple means just on slide you can see simple mathematics. Explain, for example, your one protein is phosphorylated to B protein. So what if you want to calculate the rate of change, so you can calculate it by applying the differential equations. So by simple example you can do dA by dt with respect to time, what are the changes. So it will be the productions minus degradations, or phosphorylation minus dephosphorylations if we talk about phosphorylations, right? If you talk about if A is converted into the B, so you can calculate it production minus degradations. So you can see that if you want to calculate the change with respect to a concentration, so you can, this is a constant, minus k1a plus k2b. Now question might arise in your mind, why it's in minus sign. So minus is there because a is converted into a b, so concentration of a is decreased, that's why there is a negative sign. And concentration of b is increased because a is converted to b, so there is a positive signs. And if you want to calculate change with respect to b, what is the changes with respect to time in the concentration of b, so you can see that here is k1a, because now B to A, we are talking about B to A. It can be A to B, B to A as well. So you all knows there is a phosphorylation and dephosphorylation process when we talk about proteins. So K1A, and here is minus is for the B, because B concentration is decreased because B is converted to A. So it's a simple mathematical equations. I just want to give you an idea about how these things are working behind the computers, right? So when we talk about model, as I said, we are creating the model, we are giving the simulations, and then we are predicting something, whatever you want to predict. Here we want to predict possible mechanism of the drugs. So if I give you the conclusion of network pharmacology, if you ask me by applying the network pharmacology, what we will get, if anyone of you ask me कि by applying network pharmacology what will be the output. So my answer to this question is, by applying the network pharmacology you can predict, what you can predict, you can predict possible mechanism of whatever the molecule you have. As I said, molecule can be your herbal molecule can be your new chemical entities, molecule can be a drug repurposed molecule, whatever. By you can predict, by applying these techniques you can predict possible mechanism of these compounds in xyz disease, right? Any questions on up to this? So another things you might heard about qsp, that is we call quantitative system pharmacology. So when we talk about models or quantitative, we talk about quantitative system pharmacology instead of network pharmacology. So what is quantitative? So don't be confused between quantitative system pharmacology, PBPK modeling. You know, you might heard about PBPK modeling as well, and network pharmacology, system biology, right? Network biology. These all disciplines are interconnected with each other, but there is a differences. So you should understand the differences, and then you can think of according to your research area which one I have to be applied and why. So quantitative system pharmacology, when we talk about, we will do separate session on qsp as well. It uses the mathematical computer model to characterize biological system, disease process, and drug pharmacology. It's a powerful convergence of biological pathway, pharmacology, and mathematical models for the drug development. So that is a qsp. If you want to understand qsp with respect to very simple examples, you can see on the screen, this is just a sandwich, right? You all may be using it in your breakfast, or all of you have used it. So by simple understanding, if you want to know how to measure how many calories in the form of carbohydrates, proteins, and fat, so how you can do it by these approaches. So by observing how sandwich, they can calculate the average rate sandwich enters in your stomach, right? So for example, if the sandwich is 400 calories, right, and you eat in 20 minutes, so by applying the simple mathematics you can calculate it. The input rate is 400 calories divided by 20 minutes, so it's a 20 calories per minute. Is it very hard mathematics, right? So of course basis is that one only. When you are applying with a complex case studies, then it will become complex, but basis is that one only. So 20 calories per minutes, right? So from there, and which is broken down into the individual nutrients. So every one of you are from the biology field, so you knows that when you eat any sandwich, so it will be enter into the stomach, then it broken down into the individual nutrients, then it empty into the intestinal tract and absorbed into the bloodstream, then delivered to the different organ for the utilization, storage, excretions. So every one of you knows this process, right? So if you wanted to predict how quickly your body might process a sandwich. So they need only to express each small step as a mathematical formula, and you can do it. And then you combine them into one comprehensive descriptions, that will be called system of equations, and you can do it. Are you getting my point? So mathematics can describe what is going on around us and within us. So why I am emphasizing on mathematics again and again, just tell you about, for all these calculations, models, whatever we are doing, we are talking about machine learning, the AIs, mathematics is involved. And I am also not communicating you to learn the mathematics. I am not asking you to do that, but you should have a understandings about it, what's going on, right? So by as you all maybe from non-medical field, from the medical field, not from the non-medicals. So what you should understand is how these predictions are performing by the computers, how models are being created, and how accurately they are predicting the particular things. Another things, when we talk about these network techniques, another thing is pbpk. PBPK, can anyone put in the chat box what is PBPK? Expand the PBPK. Anyone put in the chat box what is PBPK? Just expand the PBPK. Anyone in the chat box immediately. What is PBPK? You might heard about PBPK. PBPK is also very important when we talk about network, system biology. You know, anyone who can expand it. Someone is saying protein binding. Someone is saying pharmacokinetics. Yes. PBPK full form. Gaurav, you are writing pharmacokinetics. Yes, it's related to pharmacokinetics, but I am asking to explain it. Yes, Ansari Iraq 888 is correctly saying physiological based pharmacokinetic models, right? Physiological based pharmacokinetic. Isa Garg is also saying right. So PBPK modeling predict the pharmacokinetic outcome in the patient populations. So there is a word physiological based. So when if any one of you have done this modeling, you will understand that when you are doing it, you will understand that they will ask you about add the physiological parameters, and on the basis of physiological parameters, it will predict your pharmacokinetics, right? Got it? So, anyone of you heard about open system pharmacology? So, open system pharmacology is the one of the freely available, I would say, software by which you can do this PBPK modeling. Why I am saying PBPK is also important when you are going for a network pharmacology, because you all knows before pharmacodynamics, pharmacokinetic is important. If your drug is not reaching at the site, so how? What is the purpose of pharmacodynamic? Getting my point? So there is a, if I show you, if my Google is working correctly, I can show you there is a open system pharmacology. Of course it's not the scope of this, but I am just showing you, if anyone of you is interested for this, so you can just type in a Google open system pharmacology. Open system pharmacology, just add open. Is my screen is visible? Can anyone confirm it? I just open system pharmacology. So and it's a freely available. It's not asking you to give the money or anything. It's freely available. So by which you can do PBPK, quantitative system pharmacology work. You can install it in your laptop or in your systems, where you can explore it. User guide is also available. In maybe in upcoming weeks, we will also conduct the workshop. I will try to conduct the workshop on this PBPK and QSP, quantitative system pharmacology. where you will learn. Because why I am saying it's also important, it's and when you are combining PBPK with network pharmacology, and then network pharmacology with molecular docking, and then molecular dynamics, means it's a complete set where you can explore the things. Are you getting my point? Right. Okay, great. So PBPK, you can do by this open system pharmacology, right? Coming back to the slides, so you can predict pharmacodynamics, clinical efficacy outcomes in the patient population. So when you are using these open these PBPK modeling, so it will ask you if you want to see particular pharmacokinetics in particular type of patients, so you can predict it, because where you have to be put the input physiological based informations, you have to put it, and based on that it will predict it, right? So now I hope you understands about what is system biology, what is QSP, quantitative system pharmacology, what is PBPK modeling, what are the models, and how we are predicting something by using the models. So models, what is the purpose of model? Whatever the models we are creating with the help of computers, we are using the model for the predictions, right? To predict whatever the data today we have, on the basis of that I want to predict something. Like if maybe someone of you if working with respect to pharmacoeconomics, so in pharmacoeconomics also we are creating the models, Markov model, decision tree analysis, and by these models we are predicting whether after 10 years whether this medicine is cost effective or not, right? So when we are talking about model, there are three things which is very common for all types of model. One is variables. And what are variable? Variables are the things which change over the time, right? Parameters, things which do not change. Parameter will be same, right? Temperature is same. If we another kind of models, when we talk about molecular dynamics, your temperatures, your pH, and so on. So things which do not change, and functional form, which will tell you about the relationship between the two things. So these are the models. But coming back to network, now you should understand about what you all knows whatever the drugs we have today. We have a drugs which are discovered based on single drug and single target concept. For example, if I talk about atenolol, so atenolol is going to inhibit beta 1 receptors present on the heart, right, for the cardiovascular problems. So we have many drugs on single drug and single target concept. Now question arise, so why we are now talking about single drug multiple targets? Why we are talking about it? And network pharmacology is based on that, system biology is based on that. If whatever we have discussed in the first slide, I have told you, system biology means we are not studying one gene one targets, one gene one proteins. No, we are studying all together how they are interacting with each other. But now my question to all of you is, but we have practically, if you see, uh, we have a lot of medicines which are available in the market based on single drug single target concept, where we have identified a single target. As I given you the example of atenolol, there is a captopril, that is a ACE inhibitor, angiotensin converting enzyme inhibitor. There are so many examples, right? And we have a drugs in the market and we are using it for various disease. So now why we are moving towards single drug to multiple targets? Can anyone answer this question? Because network means we are not talking about single drug, single target. We are talking about multiple targets. I hope my question is clear to all of you. Can anyone put in the chat box? मेरा क्वेश्चन ये है, अगेन आई एम रिपीटिंग, कि हमारे पास जो दवाइयाँ हैं मार्केट में, जो भी हम लोग यूज़ कर रहे हैं, वो किस कंसेप्ट पे बनी हैं? वो बनी है कि हमने पर्टिकुलर टारगेट को इनहिबिट किया, या एगोनिस्ट दिया, या एंटागोनिस्ट दिया, या इनवर्स एगोनिस्ट दिया, या पार्शियल दिया, वट, तो उसके बेसिस पे हमारे पास ड्रग्स हैं मार्केट में और यूज़ कर रहे हैं हम लोग। पर अब जो हम बात कर रहे हैं बाई सिस्टम बायोलॉजी, बाई नेटवर्क, हम एक टारगेट की बात थोड़ी कर रहे हैं उसमें। अगर बनेगा तो नेटवर्क थोड़ी बनेगा। Isa Garg is saying because of resistance of disease, adapt another pathway if we block. Okay, okay. And anyone who more about these questions? I hope first सबसे पहले तो the question should be clear in your mind. मेरा क्वेश्चन अगेन मैं रिपीट कर रहा हूँ, माई क्वेश्चन कि वी हैव ए ड्रग्स इन द मार्केट विच आर बेस्ड ऑन सिंगल ड्रग सिंगल टारगेट कंसेप्ट, दैट इज द वन थिंग. नाउ माई क्वेश्चन इज व्हाई वी आर single target to multiple target. So the by the ESA is because there is a resistance. Okay, that's fine. Any other explanation? Okay, Rani any do 39 mentioning about because protein that are targeted by drug that work in. You are right, ma'am. Avdesh Infotech is saying reduce drug interactions. Okay, improve therapeutic effect. Okay, fine. So I think what my question is, yeah, you all have answered correctly that why we are moving towards it. The reason is, so do you think we have any medicines in the market and in their leaflet it mentions this drug is not going to produce any adverse effect? Do we have any drug in the market with zero adverse effect, with no adverse effect? Or many medicines have a leaflet, and when you open the leaflet in ADR sections you came to know that they have mentioned that no adverse effect has been observed with these medicines. You think there is any medicines? Yeah, so Subham is also saying one drug multiple benefit. Yeah, you are right, one drug multiple benefit, where we are talking about drug repurposing. Avinash is due to mutation of protein. Okay, with the use user tag 65 is saying for repurposing. Yeah, right. So what I am trying to put basic concept in your mind. Okay, I have created this atenolol to block beta 1 receptor on the heart only, but do you think practically when you took the medicines is it going to interact with one target only in the body? Do you think that it's going to interact with one target only? For example, आपने एटिनोलोल ली, do you think it's going to interact with beta 1 receptor which is present in the heart only? No. Yeah, Shraddha is saying no, it's not going to interact with one target. When it's going to interact with other targets, then there are two things can happen. One is, as someone of you have highlighted, Avinash has said drug repurposing. Subham is saying one drug multiple benefit. Users tech 6 is saying for repurposing and so on. So there is a one possibility is when drug is interacting with other targets, so it could produce beneficial effect. That's the one thing. But do you think it will always, when drug is interacting with other targets, apart from with this example beta 1 receptor, if it's interacting with other target, it will always produce the beneficial effect? Raina Naidu is saying screen is not visible. Okay, okay, thank you. Let me share it again if it's not visible. I'm sharing it again, so please confirm it. Now, is it visible? Now, is it visible? Raina, Rani, Rani into Neha is saying yes. Okay. So now, what I am saying is, when you have discovered the molecule to interact with one target only, so it's not necessary one target is going to interact with the other target. When it's interacting with the other targets, there are two possibilities. One possibility is by interacting with the other targets, it can produce benefit effects, right? And another scenario is it can produce adverse effect as well. Are you getting my point? As Manoj is saying yes, but not proper. Now, our is Gavarem Ray Manoj. Now, ma'am, is it okay? Shraddha is saying yes. Yeah, yeah. Subham is saying yes. So now you are getting my point. It's not only drug you can think of when drug is interacting with the other the beneficial effect, then case you can think about the drug but by interacting with the other targets it can also produce undesirable effects, and that we call adverse drug reactions. So whatever the drug we have a market in the market based on single drug single target concept, they are associated with adverse drug reactions. Drugs scientists are repurposing for the other indication as well. Why they are repurposing or why drugs are associated with adverse drug reactions? So what does it indicate? It indicates that not interacting with the single target, it interacting. That is the origin of your network, right? So as I said, when you are working with network, so when any one of you is planning to work with respect to network pharmacology or network biology, as I said, first create the network for the disease pathogenesis. That is very important, because unless and until you don't understand the pathogenesis of disease, seen many researchers who are working with respect to network pharmacology, they are not concerned about the disease pathogenesis, disease network. They are performing step by step, and then they are merging it and concluding the something else. Are you getting my that what problem you will face if the expert in the field is analyzing your result? They will ask you, computational techniques are always criticized by the experimental researcher. They will say, oh, this computer is, we are not believing on the computers. Whatever you are getting, okay, but we are not believing on it. So how you can create the confidence among them? You have to use these techniques intensive way, so that you can't include all the targets which are not relevant. So that's why very important when you are performing the steps, what should be the cutoff, what should be the threshold, what target you are getting, what is their role in the pathogenesis of the disease. Unless and until you don't understand that, you can't defend your research in the front of experts. So that's why I always first understand the pathogenesis. And when we talk about pathogenesis of the disease, any disease, and most of the diseases which are associated with lifestyle disorders, like many of you are working on, maybe working on diabetes, hypertension, and so on. So if I ask you why there these disease we are not we have, can anyone answer this question? Can anyone answer this question? Can anyone answer this question in the chat box? My question is कि हमारे पास, अगर मैं हिंदी में बताऊं, कि हमारे सर्च में किस चीज़ की कमी है, जिनके पास अच्छी फंडिंग है, एडवांस से एडवांस सारी चीज़ें एडवांस हैं कि आप मॉलिक्यूलर लेवल प्रोटीन लाइफ लॉन्ग पड़ेगी। वो अगर आपने दवाई बीच में तो क्या होगा, वो फिर ब्लड प्रेशर बढ़ जाएगा। डायबिटीज़ की दवाई ले रहा है मरीज, अगर उसने डायबिटीज़ की दवाई बंद कर दी फॉर एग्जांपल, उसकी डायबिटीज़ फिर बढ़ जाएगी, उसके ब्लड। मेरा क्वेश्चन यही है आप सबके लिए कि इतनी एडवांसमेंट के बावजूद हमारे पास कैन एनीवन पुट इन द चैट बॉक्स? वैसे स्लाइड में भी लिखा है। ओके, Isagar is saying voice is breaking. I don't know it from my side or yours. Okay, because of buffering. Understands. Not understand question. Okay. Voice is breaking. Okay. Voice again break हो रही है. Now is it clear? Can anyone confirm it? मैं फिर इसको स्लाइड मोड पे नहीं कर रहा हूँ, वैसे ही रखता हूँ. Is my voice is clear? Gaurav, Isoker 4336. Okay, it's clear. So again, I am repeating. Isagar is saying here. My question is again to all of you is कि इतनी एडवांसमेंट के बावजूद भी हमारे पास एलोपैथी में मोस्ट ऑफ द डिजीज में क्योर क्यों नहीं है? आपको भी पता है, अगर आपके घर में किसी को डायबिटीज़ है या हाइपरटेंशन है या कोई और डिजीज़ है, मोस्ट ऑफ द डिजीज़ एक बार मेडिसिन शुरू हो गई तो पेशेंट्स को वो पूरी लाइफ लॉन्ग खानी पड़ती हैं। अगर उसने मिस की तो वो ब्लड प्रेशर फिर बढ़ जाएगा, अगर उसने मिस की फिर वो ब्लड ग्लूकोज फिर बढ़ जाएगा, उसकी डायबिटीज़ कंट्रोल नहीं होगी। इवन दो इन आयुर्वेदा मैनी आर सेइंग वी विल क्योर इट, बट देयर आर मैनी एग्जांपल patients are switching their medicines from allopathy to ayurveda and still they are not getting the cure. Their blood sugar is again increased, not controlled, and then they go back again to the allopathic medicines. But allopathic medicine, whatever you are using it, you have to use it on regular basis. So my question to all of you is, even means we have lot of advancement in the field of science, why don't we have a medicines for the cure? Allopathy goes for symptomatic relief. Now, yeah, Subham, you are saying right, but why? Why not for cure? Subham Tomar 6026. Why not for cure? Subham, am I audible? हेलो यार, बफरिंग फिर होना शुरू हो गई लग रहा है. Me move forward. So answer to this question is, the one of the reason is, yeah, Rani is saying right, because most of these chronic diseases are related to genetic illness that cannot be targeted by the medicine. Okay, but now we are targeting the genes as well. We are targeting these one as well. We don't know what are the, yeah. Isa Garg is saying right, because we don't know the exact reason. The student, in etiology sections, you can write the exact etiology of this disease is unknown. That means it's idiopathic in nature. Pollutions, heavy metals, so and so on, and genetic factors are playing a major role in the development of these diseases. So unless and until if we don't know about the exact reason for the disease, so how we will cure the disease? We are not able to cure. We can manage it, as Subham is saying, because our purpose is to manage. Why we are focusing on management? Because we don't know the exact reason, and unless and until we don't know the exact reason, how we will cure the disease? But in some kind of disease like infectious disease, For example, you got a bacterial infections. So do you have to take antibiotics for your entire life? No, right? So you have to of the treatment, and there is reason, because this is the, so you will say हो सकता है pollution की वजह से हुआ, but if someone is in Himalayas that is also getting cancer, so you will say maybe genetic factors. So you are predicting ki genetic factors, maybe this factor the interacting, and it result in the development of disease. So due to, because we don't know the exact etiologies, we are not able to cure. Are you getting my point? So here is the network which can help you to understand the pathogenesis of the disease in a better way. To but first network which is we are dealing with pathogenesis of the disease or drugs, understand the pathogenesis of the disease, understand the signaling pathway, intracellular signaling pathway which are involved in that particular disease, then only you can do network, or you can apply system biology, QSP, PVPK, all these things in a better way. And this is the root cause problems which you will see in the network projects which students are doing. They are not worry about, they are saying नो नो हमें बस इतना बता दो क्या क्या करना है, कहाँ पे क्या डाटाबेस है, हम डाटा पुल आउट करके उसको बना के खत्म कर देंगे. That is not the right way. And when you ask the question to them, they are not make you understand, because they have not read about these pathways, these proteins, these genes, these metabolites. So how you will defend it, right? So that's why I am saying first part we will focus on biology, these disease network, then we will uh, go into the network pharmacology. So now I hope every one of you get a sense what is network pharmacology. Network pharmacology, because word pharmacology is attached, so when pharmacology is attached, we talk about the drugs, right? So how drugs can interacting with the multiple targets. And as I said, when drug is interacting with the multiple targets, there are two possibilities. You can identify the molecule for the drug repurposing. Are you getting my point? You can identify the possible adverse effect of the drugs as well, right? So these, so one of the area where you can apply the network pharmacological technique is drug repurposing. And how you can identify the drug repurposing molecule? By understanding the interactions of the drugs with uh, with the other targets. We call it off level targets, right? Are you getting my point? Any questions so far? So network is, now I can't say it's a emerging discipline, it's a useful in the drug discovery, and it combines genomic technologies, system biology, chemistry through computational biological tool. So that's why I am saying, if you are very good in the computers, but if you are not good, or you don't have a good knowledge about means biology, so how you will do the network pharmacology in a better way, right? You can't do it. So biological network, human bodies at the steady state and natural equilibrium state is at the healthy state. So first you have to understand about how proteins, how metabolites, how these signal transduction networks are, these all with each others in a healthy individual. You know, in clinical trials, you always have a comparator, right? You have a comparator with placebo controlled, you have your comparator can be active comparator, and so on. So unless and until if you don't understand about signal pathways by which our body working, by which healthy body is working, so how you will understand what alteration is happening in the disease state, and now how your drug can modulate that. There are three aspects. One aspect is you should understand about interactions among the proteins at the healthy state individual, in healthy individual. Just one second. Am I audible? No, not yet. Okay. So, first you have to understand about disease, stable network with a specific topological structure and certain network properties. And as I said, first you have to understand that what are the how proteins, metabolites, all these are interacting with each others in normal states. And then you have to understand about what happening when there is a when these networks become unstable with different topological structures and network properties. And then you can understand about the network pharmacology is the based on theory of network biology and biological balance, and provide a new ideas for the drug discovery as well as for understanding the mechanism of functioning. Am I audible, or is my screen is visible? Yeah, someone is saying, is it possible? But there are a multiple factor are responsible which pathway should be selected, and go ahead with one time, not a single drug which can go for all related pathway. Yeah, definitely. Voice not clear. Voice is not clear, Subham. So with yes, unaudible. Okay, let me share it again. Very difficult to conduct. Now it's clear. Okay, great. So I hope my slides are also visible. Yeah. So now I am not going into the pharmacology. Every one of you knows that there are two parts. One we called drug actions and drug effect, and particularly drug action is something which is not visible. You only can see the effect. If, for example, you got a headache, you took the medicines, so headache if disappear, you can see the effect only. But behind that, what are the possible mechanisms? What are the possible signalings which are hampered? You can't see that, right? So that's why we call drug actions and drug effect we all studies in the pharmacology. And these are the different types of receptors. I am skipping that part because everyone of you knows about these. But again, I request every one of you, whenever you got time, go through these all, their signaling, their pathways, and particularly with respect to your disease, on particular whatever the disease where you want to apply your network techniques, read much details about mechanisms about that particular disease, so that you can later on when you apply these techniques you can understand better, right? So network pharmacology is combined mechanistic model of disease pathophysiology, PKPD of a therapeutic targets, and quantitative experimental data. And whatever the models will results can be used to run simulations to understand how drug modify cellular networks, how they are impacted by the pathophysiology, the significant pathways, the impact of drug parameters, and biological variance on drug efficacy and safety, right? So starting now, we are entering into the network. So we are going to start with the basic networks. What is the basic networks? How you can deal with the basic network? But before that, if anyone has any questions, so now we are entering into the network. So before network, I think you can understand better by the social network. So every one of you know social network. There are a social website as well. You are talking about Facebook, Instagram, Twitter, and so on, right? So in these, there are everyone is connecting with number of individuals, right? So this is also the one of the network, and these kind of network we call social network, right? So I think in these era, every one of us is using these social networks, and now you can see that there are a lot of connections. This one friend is of mine, then he is also my friend. He and she is also the friend. There is a mutual friend. There is other friends. So there is a lot of connections. So in network, in simple network, these circles we call node, and the lines which are connecting to one entity to another entities, that we call lines or edges. So when we talk about network, there are two things. One is you heard about nodes, or we also call it vertices. Some researcher are using vertex, vertices word. Vertex is the one. If there are a more than vertices, then nodes, node or nodes, right? So node, this is node, and this node is connecting with another node through the lines. So these are the lines, or we also call it edges. So line or edges, we represent it L, capital L, or E. So both are same things. Are you getting my point? Anyone has any questions? So we are considering about the social network, so but of course we are not from the social background, so we are not dealing with the social network, but the social scientists are working on these social network. And if you want to identify, if someone has done some mess up by using social website or something, by network analysis you will get to know that how these connections are connected. And when you will reveal that, you will get to know that how, what are the means different layers in that, that is the social networks. There are a, this is again the example of social network, where you can see that these are the direct friends. So someone is of you is of direct friend, someone is not the direct friend, what are the mutual friends. This and this one is not the direct friend, but there are a mutual friends. And you all knows, when you are using these Facebook or other things, you will find that their statistics is showing that 20 mutual friends you and this person has, right? So that is a network. By studying these connections, they will get to know. And there is a indirect friends as well, there, that is represented by the green line. So all these analysis we are doing with respect to social network, right? So now when we talk about network, so as already explained you, social networks like this Facebook, Instagrams, but there are a not only the social network, there are a airport networks, right? Sometime question arise in my mind ki jo aeroplanes, why they are not colliding with each others. How they get to know that this is the path, this is the where they has to be go, what are the different networks. So there is a lot of airport network as well. There is a road networks, right? Highway networks. So how roads are connecting with each others, where they have to connect, how to connect, what is the hub, right? So that we talk about road networks. We talk about brain neural network, you might heard about. That is a complex network where number of neurons, the word is coming from the with respect to brain, because neurons, there are a huge number of millions of neurons, billions of neurons, right? So they are interacting with each others and connecting with each others and producing the signals. So of course if you go through the literature, you will find that these network techniques are also being used by NASA, also used by ISRO, also used to identify the terrorist as well, right? You know all about the Operation Sindoor. In Operation Sindoor, Indian government has or Indian armies or forces have, Indian forces have attack only on particular hub, where they got to know that there are a terrorist hub only. So how they have identified, if you go through the literature, you will find that these identification is based on graph theory, how to identify the hub. And in case of biology, who is our terrorist? Our terrorist is disease, the things which has caused the disease, which have done the mutations, which have done the changes in the bodies. By that one, I developed that particular disease. Are you getting my point? Any questions so far? So that's why being a biologist or pharmacologist, we are interesting about the biological network. And again, biological network are not a simple network. You all knows there are number of things, number of proteins, number of metabolites are formed. A lot of things are interacting with each others, and it's not a simple process. When we talk about biological network, it's a complexities. And another complexity is when you link it with the clinical perspective, you will find that patient nowadays is not suffering only the single disease. Most of the cases you will find in the clinic, the patient have diabetes, hypertension, fatty liver, this, that, all these combination. And when there is a lot of comorbid conditions, your network will become more complex, because one protein can have a role in the multiple disease. When we talk about PI3K, AKT, mTOR pathways, so do you think PI3, AKT, mTOR pathway is playing role only in a single disease? Can you answer that? Is one pathway is playing role in the development of single disease only? Yes or no? Put in the chat box. Subham, Isa Garg, Manoj, Kavarin, Manoj, Avinash, Avdesh, Shraddha, Yukta, Rani Naidu's, Avinash, sir. Yes. Can anyone want to answer? Multiple pathway. Yes, you are right. Multiple pathways crosstalk, right? So when the patient who have a multiple disease, their network will become more complex. It's not easy role, right? Yeah, Tanisha is saying no. Yeah, Isa Garg is saying there are affecting multiple pathway, right? Indu is also saying multiple pathway crosstalk. Yes, you all are right, right? Avinash Gunjal is saying no. Yeah, so all these network. When we talk about network, biological network, we these networks are based on the graph theory. So if you knows about graph theory, you just read about graph theory. And as I said already, this already I explained to you. When we talk about patients, so patient is suffering from a multiple disease, and when a patient is of multiple disease, multiple interactions, and it's very difficult to identify the particular targets which is playing in a role in the development of that particular disease, right? So already I have explained to you that single target to multi network modulating therapy, or single target to multiple targets. Why we are talking nowadays, everyone is talking about single target to multiple target, because now we understand that the one molecule is not interacting with one target, it's interacting with the multiple target, and due to that we are getting adverse effect. Due to that we are identifying the potential molecule for the drug repurposing. That is the basis, right? So whatever the drugs we have identified earlier, now we came to know that yes, these drugs are not interacting with one target, they are interacting with the multiple targets. So network pharmacology, this already I have explained you. So importance, that is another questions which beginner always ask, where we can apply these network techniques, sir? Where we can apply these? I am from chemistry background, so can I use network pharmacological techniques? I am from pharmacognosy background. Can I use network techniques? I am from pharmacology background. Can I use these techniques? I am from PharmD background or pharmacy practice background. Can I use these techniques? Yes, all of you can use these techniques. Now, another question is, you know, I'm just putting question to all of you is, for example, someone comes to you and that person is saying to you, I want to apply network pharmacological techniques, please help me. And you ask where you want to apply. That person said, I want to identify, I want to uh, for example, if we talk about drug repurposing, so they are saying, okay, I want to repurpose ACE inhibitors for epilepsy through mTOR, right? Again I am repeating, someone comes to you and saying I want to use network pharmacological techniques to check effect of uh, any drug, for example ace inhibitors in epilepsy targeting mTOR. mTOR, you all heard about, mammalian target of rapamycin, right? So now tell me, is it a good topic for network pharmacology? Yes or no? And if you are saying yes, then why? If you are saying no, then why no? Put in the chat box. Also give the explanations. Again I am repeating, ace inhibitor, someone comes to you and want to apply network pharmacological techniques where they want to check effect of ace inhibitors, means they want to repurpose ace inhibitor repurposing for epilepsy targeting mTOR. Is it a good topic for network? Yeah, Israel Koro 8617 is saying, yeah, we will take all the queries. You, every one of you can put your queries in the chat box at the end. I will take all the queries. So you continue to put Isar Lokoro 8617. We will take the all the queries individually as well. So you just wait, right? But now answer my question. Avinash is saying yes. Why yes? Also put the explanation. If someone is saying no, then also put the explanation. Okay, if someone is saying no, they can put the explanation. My again question is, if you want to, I want to repurpose ace inhibitors for epilepsy targeting mTOR. Is it a good topic for the network? Avinash is saying yes. What about others? Yes, put the, you know, immediately your response in the chat box. अब तक जो भी आपने समझा उसके अकॉर्डिंगली हम रीपर्पस करना चाहते हैं एसी इनहिबिटर्स को, कोई भी एसी इनहिबिटर, फॉर एग्जांपल कैप्टोप्रिल को मैं रीपर्पस करना चाहता हूँ एपिलेप्सी के लिए थ्रू एमटॉर, टारगेट है एमटॉर, मैमेलियन टारगेट ऑफ रेपामाइसिन. I am not going in detail ki whether mTOR will be a good target or not a good target, this that. I am not going through that. But my question is it a good for the network? Avinash is saying yes. Okay. Another one, Rani Indu is saying yes, this can be done by network through overlap of disease, gene target, then gene ontology enrichment. Okay, good. Then another Sobit is saying yes, it is good, but first we have to see the AC inhibitor targeting, then we have to search mTOR. Okay. Isa Garg is saying yes, sir. We can identify it if AC inhibitor can target mTOR in epilepsy if there is a common gene. Okay. Gaurav is saying I can apply network pharmacology for herbo mineral drugs. Yes, you can apply it. We can done by overlap. Yes, with the help of network, we can easily identify whether the AC inhibitor interacting with mTOR or not. Yeah, you are saying mTOR pathways or not. Okay. Answer to this question, my answer is no. Yeah, Subham is saying right. No, if drug is already okay. Subham, drug is already decided means your answer is right, but your explanation is not right. Subham Tomar. Yeah, Gaurav, we will take your question at last about the synergistic, antagonistic, or addition effect. But answer to this question is it's not a good topic. Now you will ask me, sir, give the explanation why you are saying, every one of us is saying yes, it's a good topic. If we want to repurpose captopril for the epilepsy targeting mTOR, then what is the problem? Problem is you have already identified the targets. You have already identified the target, and target is mTOR, right? Now what is the need of network where you have identified already the target that this captopril is going to produce anti-epileptic effect by inhibition of mTOR? and by applying the network, what you will get out of it? Because you have already identified the target. Are you getting my point? So this is the practical issues, sometimes student comes to me and they are saying, सर वो सर ने भेजा है या मैडम ने भेजा है, हमें भी नेटवर्क करना है, और ये हम एनिमल स्टडी कर रहे हैं, हमने एनिमल स्टडी कर ली, और ये हमारा टारगेट है, ये हमारी ड्रग है, ये हमारी डिजीज है। अब आप बोलते हो नहीं भाई, ये इसके लिए तो नेटवर्क सही नहीं है। गॉट इट? I mean to say, if already decided. Yeah, Subham, but if drug is already decided, that's fine, because drug is needed. If drug is already decided, that is required to do the network pharmacology. Drug is needed. Yeah, you are right. You are understanding that point of view. By network, we can identify the drug repurposed molecule. That is right. But if you have identified the drug as well, and now you want to identify its possible mechanism, you can still apply the network pharmacological techniques. Subham, are you getting my point? There are two things. One thing is you can apply the network techniques to identify the drug repurposing molecule. That is the one. But if you have identified already the drug repurposing molecule, now you want to apply network techniques to identify its possible mechanism, you can apply the network techniques. Are you getting my point? Again, I am repeating कि अगर आपके पास कोई मॉलिक्यूल है, चाहे हर्बल है, चाहे सिंथेटिक है, वट एवर द मॉलिक्यूल यू हैव, एंड आपने ऑलरेडी डिसाइड कर रखा है उसका टारगेट भी, उसकी डिजीज भी, राइट? आपने एनिमल एक्सपेरिमेंटेशन भी कर चुके हो, आपने आरटीपीसीआर से वो पर्टिकुलर जीन के थ्रू आपने इफेक्ट भी देख रखा है, या आपने वेस्टर्न ब्लॉट से वो प्रोटीन भी चेक कर रखा है, सब कुछ। तो उसके बाद फिर बचा क्या? उसके बाद अब आप बोल रहे हो अब मुझे थोड़ा सा वो आई एम वांट टू कि थोड़ा सा मेरा वॉल्यूम बढ़ जाए काम का, या कुछ हो जाए बढ़ाने के लिए, अगर इफ़ यू आर अप्लाइंग दीज नेटवर्क टेक्निक्स, इट्स नॉट द राइट वे, because you have already identified the target, you have already the drug, you have already done experimental part. Now what you want to get by the network? Are you getting my point? If question is our hypothesis, then we can verify by the network. Yes, uh, Rani Indu 3987 is saying right, because computational techniques, we most of the computational techniques we are using at early phases of drug discovery. Computational techniques we are using before experimental validations, right? Yeah, now Rani Naidu Indu is saying if in vivo is done, then network is required, it's pre-request prior to in vivo. Yeah, there are two things. One thing is, you know, s- with respect to Indian academic scenario, I am just putting these things. S- when we are doing network, all these things, it's very difficult in most of the institute or colleges to combine it with the experimental validations. The reason is because you have to do Western blot, and to do the Western blot or RT-PCRs, their reagents are costly. Your primary antibodies are costly. To do the Western blot, primary antibodies, right? So primary antibody is costly. Most of the time, it's means more than 25,000 approximately. So what problem we are facing, or everyone is facing, regarding the validations of the network results by experimental validations? Of course you can do it if you have the facility, you have a significant funds you have, then you can do it. But as I know, most of the institute, we don't have that much fund, unless and until you have a funding from ICMR or DSTs or other regulatory bodies. That's fine. But now maybe questions from your side is, how we can deal with this problem? Can we restrict ourselves up to network, molecular docking and dynamics, that part only, and then we submit our thesis or go for the publications. And if I am PhD student, so that is not the enough. I have to do the experimental work. Without experimental work I can't submit the thesis. So how I can deal with this problem? Is it the question from your side? पहले तो क्वेश्चन समझ आना चाहिए. यू शुड अंडरस्टैंड द क्वेश्चन. क्वेश्चन ये है कि आपने एक्सपेरिमेंटल से, सॉरी, नेटवर्क से यू हैव आइडेंटिफाइड द प्रोटीन्स, pathways, this that. Now next step is validations. Now expert will ask you, okay, by network you have identified this target and these things. Now show me experimentally. Do the in vitro or in vivo. So for in vitro is also costly process, in vivo is also costly process. So how to deal with this problem? So to deal with this problem is, solution to this problem is, first you conduct the animal experimentations. First you conduct the animal experimentations. Means if you are dealing with the pharmacology, you have animal house and you conduct the animal experimentation first. अब नेक्स्ट क्वेश्चन माइट अराइज इन योर माइंड कि सर ये क्या बात हुई कि हम पहले एनिमल एक्सपेरिमेंटेशन कर लें. अभी आप बोल रहे थे कि अगर पहले एक्सपेरिमेंटल कर चुके तो नेटवर्क का कोई मतलब ही नहीं है. अब आप बोल रहे हो कि उस प्रॉब्लम से डील करने के लिए पहले आप एक्सपेरिमेंटल कर लो फिर नेटवर्क करो. आर यू गेटिंग माई पॉइंट? आई होप आई एम ऑडिबल. Yeah, Sobit is saying due to lack of facility. Yeah, due to lack of facilities, due to, because these are the costly processes, right? Even for a single proteins, you have to spend the monies, and if it's not a research institute, or if you don't have a funding, so definitely every one of you will face the difficulty to validate it. And of course in master maybe it's okay, but when if you are doing PhD, so it's not acceptable. So to deal this problem, what I am saying is, first conduct the experimental studies, experimental animal studies if you have an animal house. Because in vitro is again costly process, but if you have an animal house, you do first experimental studies, experimental animal studies. Now question is, as I said, if you have, I have given the example, you want to repurpose captopril for the epilepsy through mTOR, or for example someone of you have already done the experimental work and you have identified all these, then what is the purpose of network? There is no purpose. You can't combine it, because if you already know the target, already you have a drugs, already you have a disease, what is the purpose? What you have, you will come out from the this one by the by doing the network. So solution to this problem is, what you can do is, for example you have hypothesis, as someone of you saying about the hypothesis, Rani Indu is saying about hypothesis. For example you have a hypothesis, same hypothesis like captopril in epilepsy, right? You want to repurpose captopril in epilepsy. So first you conduct animal experimentations, simple animal experimentations, because at molecular level animal experimentations, again cost will increase. And why you are planning first animal experimentation? Because you don't have that much fund to do the western blot, RT-PCR, and so on, other molecular techniques. So you conduct animal experimentation, simple animal experimentation, which every college can do it. You just take the drug, do the grouping of animals, and do some behavioral parameters or racine score, by which you will get to know about seizure or intensity of the seizures. And you have done these 3 4 simple experiment, by which you got to know that yes, captopril is working in the epilepsy. You just done the visual observations, make the videos, and by that one you are comparing with the control group and 2 or 3 doses of your captopril, and you came to know that yes, captopril is, from this experiment you came to know that yes, captopril is working at this dose to reduce the epilepsy. This experiment you have done. and it's not the costly experiment, right? Because you have not done any Western blot, anything else. Now, next part, you have done. Just 1 second. Hello. Yes sir. बढ़िया सर, बढ़िया. हाँ सर, हाँ. मैं पता करके बताता हूँ सर. ठीक है सर, ठीक है. मैं बताता हूँ सर. ओके. हाँ जी सर, हाँ जी. ठीक. हाँ जी सर. Okay, so what I am saying is, do the simple experiment first. Just check your hypothesis whether this molecule is working or not. Like in this example, as I said, you have checked whether captopril is working in epilepsy or not, and now you came to know that yes, at this dose, intensity of seizure is reduced by giving this captopril. Now, next part you have done network part, and now you can apply the network, where you can say that first we have conduct simple animal experimentations where we have checked whether it's working or not. Now we came to know that it's working, but we don't know the possible mechanism behind it. To get to know about the possible mechanism, we applied network techniques. And now we came to know that, because animal experimentation I have already shown you, and I have explained that yes, this at this dose my molecule is working in the epilepsy, and by the network I can show to the audience that this is a possible mechanism by which this molecule is working. And in the future perspective you can add, in futures another researcher can explore, can validate experimentally whatever the targets I have identified. Are you getting my point? Any questions? अगेन आई एम रिपीटिंग, ये हमेशा चैलेंजिंग पार्ट है, हमेशा प्रॉब्लम आती है. प्रॉब्लम ये आती है, आपने नेटवर्क कर लिया, नेटवर्क से आपने सब कुछ निकाल लिया, टारगेट भी निकाल लिया, सब कुछ पाथवे भी बना लिए. नाउ यू आर अनेबल टू कंबाइन इट विद द एक्सपेरिमेंटल वैलिडेशन, because next step is you have to validate it. Now, as I said, many experts are always criticizing the computational technique. They will say no no, ठीक है, कंपटीशन से बस कंपटीशन ने नहीं किया है, और पर्टिकुलर इन फार्माकोलॉजी, अरे एनिमल एक्सपेरिमेंटेशन वाला पार्ट कुछ नहीं किया. और अगर आपने किया नेटवर्क करने के बाद, देन दे विल आस्क यू द क्वेश्चन कि नेटवर्क में यू आर शोइंग अस दिस टारगेट, एंड नाउ एक्सपेरिमेंटली you have not validated it. Why you have not done Western blot? Why you have not done this? And you can't answer ki we don't have the facility, that's why we have not done. If you are submitting your manuscript to the international journal also, these things will not be acceptable, right? So to deal this problem, as I said, but your hypothesis would be new, novel. It's not like that. If it's already done, then question will arise why you have done the animal experimentations, right? Yeah, Subham is saying how can I say by that mechanism this effect arise? Yeah, by the possible mechanism you are predicting. By the networks you are saying that this is the possible mechanism by which this response we are getting. But of course, you are writing in the future perspective, experimental validation is required. But you will be skip from the problem which you will face, that you have not done animal experimentation or you have not done any experimental work by this way. Subham, anyone else? Are you getting my point? Your comment is not visible yet. Specific mechanism not predicted. No, no, specific mechanism is predicted, but their validation is required. By network, it's not only the one mechanism you will going to predict. You are going to predict possible 2 3 mechanisms, and by the in vitro studies, then you will get to know that okay, this mechanism playing this much role, this mechanism playing this much role. So you have to put specific inhibitors. In the presence of inhibitors, what is the effect? Without inhibitors, what is the effect? Then only you can identify the role of these things. Got it? Are you getting my point? Yeah, because experimental, that's why. But as I said, because I am also Indian and I am in Indian academic, so I know for every one of us, it's not possible to do studies at the molecular level in much more detail, because everyone has the limitations. But you should know how to avoid the limitations. You can't say no, 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 हम कर ही नहीं सकते. You have to think in a different ways how to deal with that. Another way is with respect to pharmacognosy peoples or herbals who are dealing with herbal is what they are doing is they are making the extract, plant extract, the ethanolic extract, this extract, and like that extract they are preparing it. Experimental work, I am saying. So they are doing this work first. They are making the extract of the leaves, bark, whatever, and different extract they are preparing, and then they are doing analytical techniques by which they identifying by HPTLCs or LCMS-MS. They are identifying the phytoconstituents or most bioactive constituents in that particular extract, right? And after identification of these constituents, they are applying the network techniques. That is another way to couple experimental techniques with the network. Pharmacognosy people are there, the researcher who are working with herbals. So how you can couple your experimental part with the network part? You prepare the extract, right, of whatever the plant you are selecting. Prepare the extract, identify the bioactive molecules or their phytoconstituents by using any analytical techniques in which you can identify it. And after identifications, you can apply the network. Now you have a these are the major bioactive present in this one. Now you apply the network. So your experimental part plus network part, and then you can get good project as well and good publications as well. Yeah, is Israel Lekoro 8617. You are from herbal field, okay? So you can apply in that way, right? Israel Lekoro 8617. You can apply in that way for your herbals, and of course, in similar way, you can do experimental part as well. You mix the extract, do the simple animal experimentation, and then you can apply it network, right? Avdesh Infotech is saying how we predict particular pathway from multiple pathway to target in the experimental studies. That we will cover tomorrow as well. How to create it, hands-on sessions, and how you can predict particular pathway is playing a important role when you we are doing the ranking, right? So what I am saying is in which area first you understand in which area you can apply the network techniques and how you can couple it with the experimental techniques. That's very important. Yeah, impact database where you will get a lot of herbals molecule structures are there, right? Indian medicinal plants databases. There is a marine databases as well where you will get marine compound structures. But again, to couple it with the experimental, first you do some experimental part and then you can couple with the network. Otherwise again you will face the problem. When you are submitting to the journal, they will ask you no, 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 that is not enough. Give the validations, experimental validation results as well. Okay, so we will take the question at last as well. Someone is asking some question, but I think it disappear. I'm just reading that questions about some extract or whatever. So again, in this slide, you understand that where you can apply and where you can't apply the network techniques. Where you can't apply, where you already knows the target, you already knows the disease, you already knows the drug. So what is the purpose? देन यू विल से नहीं सर तो मना कर रहे हैं कि इस पे नेटवर्क नहीं हो सकता, बट लिटरेचर में तो लोगों ने कर रखा है. बट यू हैव टू अप्लाई योर माइंड कि if you already knows the target, already the drugs you have, already the disease you have, so by the network what you are going to get, right? You can say that okay, I want to see other target, but whether you can other target you can explore it experimentally, that is not possible. So, identify if you want to identify potential drug candidates, you can apply these techniques. You want to understand the mechanism of the drugs, you can apply these techniques where you can talk about new chemical entities, whatever. If you want to understand the possible drug-drug interactions, you can apply these techniques. If you want to identify possible mechanism of adverse drug reactions, that can also where also you can apply. We are applying currently. We are using network techniques to identify the possible mechanism of adverse drug reactions, because our lab is working on signal identification as well by using Bayesian and frequency based approaches. So by identifying the signals, because it's not already known, we are coupling it with the network techniques where we want to understand what is a possible mechanism, and after identifications you couple it with the molecular docking and then dynamics, molecular dynamics, then it will become a complete set, right? So this is the area where you can apply it. And one, if someone is again you can put the question someone is putting, I just read, not means read completely that questions, but that question is something about the extract or something about marine, I think so. So you can put it again. So this is the importance. And if we talk about popularity of network pharmacological techniques, you will find that if you just do the PubMed, you know, you will find that how popular these techniques are. These techniques are very popular. Any questions so far, or I can take the questions after these slides. So you can see that popularity of network pharmacology, just put in the PubMed in the search network pharmacology, you will find that huge number of studies and significantly increasing over the years. And if you do little bit more search, you will find that lot of techniques, these techniques are being used by the Chinese researcher. And when you read this paper, you will find that they are using these techniques for the traditional Chinese medicines that we call TCM. Traditional Chinese medicines, why they are using now, next question arises, why Chinese researcher are using these techniques for the TCM, traditional Chinese medicines? So you will find that traditional Chinese medicines are somewhat similar to our herbal molecules, right? So herbals are always criticized at the global platform for their lack of scientific evidence. You know, we talk about okay, this herbal is effective in this one, this is effective, and when at the global platform they will ask you, show me the evidence, show me the data which says that this herbal is effective in this one. And we don't have a proper conducted clinical trials. And of course, to do the clinical trial, it's a huge expensive, every one of you knows that how expensive the clinical trials are. So sir, what the traditional these Chinese researchers are doing, they are generating the scientific evidence by using these network techniques coupling with the experimental techniques. Are you getting my point? I think Sobit is saying, kindly repeat regarding adverse drug reaction techniques used by your lab. That is a signaling. Yeah, Sobit, actually, what we are doing is we are identifying the novel signal of the drugs. Novel signal of drug means the adverse drug reactions which are not earlier reported or observed in the clinical trial, that is we call unlisted adverse drug reactions. We are identifying these signals, and because these are not earlier known, their mechanism is also unidentified, then we are coupling with the network techniques. Yeah, mommy tare. Okay, we will take all the questions at the end of sessions. What I am saying is, in this slide is popularity of network. So popularity of network, just search in the PubMed, just add network pharmacology. You will find that lot of scientific evidence or scientific papers are there, and every year their number is increasing significantly. So if you do little bit more research on that, You will find that, open this paper, you will find that most of the papers are being published by the Chinese researcher. Then open these paper, then you will find that they are using network pharmacological technique to generate scientific evidence for their traditional Chinese medicines, TCM. In China's, first treatment to the patient is given by TCM, and if you see the composition of TCM, you will find that that is somewhat similar to herbal molecules. Are you getting my point? So now they are generating, and as I said, because herbals are always criticized for lack of scientific evidence, so they are generating scientific evidence for their medicines. So now question arise in mind is, why not India? Why not Indian researchers? Of course, some of researchers are using these techniques, but by conducting these kind of workshop, I hope many of you will also start working on these areas and explore it and generate the scientific evidence for our herbal medicine as well. Question is, why not India? India have a rich heritage of our herbals, we have lot of scientists. If you every time if you see top 2% scientist list, you will find many Indians are there, and Indians are very good in programming or computational things. Indians are very good at, so the most of them are Indian only, kind of things. So this is the questions which arise in the mind, why not India? I hope now again it's blurred. Am I audible? आप कॉलेज में हो? अच्छा अच्छा, that's all. Am I audible? Kindly repeat regarding, I think again problem is there. From which data date? No, sir. क्यों नहीं चल रहा? हम्म. हाँ, तो हो रहा था. Am I audible? अब क्वेश्चन ले लेते हैं. आई थिंक वो स्लाइड्स में कुछ प्रॉब्लम आ रही है. uh, you can put your queries in the chat box. I am going to read your queries in the chat box. फिर स्लाइड दोबारा स्टार्ट करते हैं. कैन एनीवन कंफर्म इट? एम आई ऑडिबल? पहले तो ये कंफर्म कर लो. Slides are not visible. Slides, I have just stopped. Rani, just let me know, am I audible? So meantime we can take the your queries, and then I will move forward. Okay, great. So okay, great. So let me read your queries. You can continue to put your queries. I am going to check chat box from starting. When started? Okay. Good morning. Screen is not visible. Screen is not visible. Screen is not visible. Voice is breaking. Screen is not visible. Voice is breaking. Same issue, sir. Voice is in. Screen is not visible. Screen is not visible. Voice. Your voice is not audible. Breaking. Please work on the screen. Visibility. Voice. Visible. All clear. Yes. Yes. Please go ahead. Shark cry. Screen is not clear. Screen is not visible. Yes, sir. Your voice is clear. Yes, voice. Okay, now it's clear. Screen is visible. Screen is visible. Screen is clear now. Clear now. It's loading. Buffering. You are under poor network connections. Yes. Buffering. Loading. Now it's okay. Yes. Continue to put in your chat box physiological based pharmacokinetic. Kinetic. Yes. Ansari is saying right. Physiological based pharmacokinetic modeling. We called. Isa is also saying right. Pahari focus physiological based pharmacokinetic. Subham, you all right because of resistance or disease. Okay, these are the answer you have provided right. Drug repurposing mutations. Okay, screen is not visible. Yes, 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 yes. Your voice is break. Yes. Allopathy goes for symptomatic relief. Okay. Genetic, but there are multiple factors which are responsible. Okay. Questions I am not getting in the chat box. I do, yeah. One question is Israel Lekoro 8617. Are you there? Your question is I do natural product chemistry and identify compound through GCMS. Can it be applied to network pharmacology? Yes, why not? You can do that. You can do that. I think Israel, as I answer you in earlier as well. So one way is you identify the natural compounds, you do these analytical techniques, GCMS, LCMS, whatever, HPTLCs, and then you couple it with the network pharmacology where you can identify the possible mechanism of your identified natural compounds in particular disease. Israel, is it okay to you? Right then, and of course, for every one of you, I would say that after network you will get to know about possible target, but with the targets how your compounds are interacting in the active site. To get to know about that, you couple it with the molecular docking. So molecular docking tell you about energetically favored binding conformations of the drugs or molecule in the active site of the target, because network will not tell you about that. Network will tell you about how what are the possible targets which your compound can hit in that particular disease, but how your molecule is interacting in the active site you will not get that by the network. So to know that, you couple it with the molecular docking, and whether your molecule is stable in the active site, you get to know it by the molecular dynamics. Got it, Israel? Now next question. Yes, this can be done by network through overlap. Yeah, Rani is saying of disease and gene target and then gene ontology enrichment. Yes, yes, it's good. But first we have to see the ACE inhibitor. Okay, that is the answer which you are providing. Yes, with the help of network, we can easily understand whether the IC inhibitor is interacting with the mTOR pathways or not. No, who is this? Avinash. We can't say that it's interacting with the mTOR or not. To check the interactions, you have to do the molecular docking. Right? No, if already knows. Okay, I mean, if the question is our hypothesis, yes. If in vivo is done, then network is not required. It's a prerequisite. But as I told you, you can couple it. But how you can couple it? That already I have explained you. You can do simple animal experimentations, but your hypothesis would be new, and then you can apply the network. How can I say by this mechanism the effect arise as there are multiple mechanisms? Yes, of course there are multiple mechanism, but which network, by analyzing the network you will get to know that which network is playing a significant role in the which protein is playing a significant role in the development of disease. And it's also happen for one disease, maybe pi3k is a good target. But for other disease, that is not the good target. That also you have to take care of when we are selecting the target. And I always say to the students, when you are selecting the target, don't entirely depend upon the ranking which is generated by the computers. Ranking generated by the computer is based on degree, or eigenvector centrality, or any other measures. So they are, computer is providing you the values based on that. But sometimes you can also ignore the first rank targets which is provided by the computers with proper justifications. Because, as I said, for example, you have selected the target, or your computer system has selected the target based on degree. And what is a degree? Degree is how many connections your particular target has. So for example, this particular target has lot of connections, so based on that I am selecting that target. So but you know that if I am targeting this target by my drug, it will interfere with the biological process as well, normal biological process as well. Because whatever the target you are selecting, you have to think about its biological function as well. If you are altering, if you are inhibiting it, it could result in various major adverse drug reactions or major problem, so that you can't be justified. So sometime, if computer is telling me this is the first rank, I can exclude it with the justification that this is the not, this could not be the first rank for my hypothesis. Because as per my knowledge, this target if I am hitting, it will create the problem, right? And you can validate it, you can go through the clinical trial, and you came to know that someone have targeted this target by the drug, and drug, that clinical trial has been suspended due to adverse drug reactions. So you got a hint that no, no, this is not the good target, even though my computer is saying this is the first rank target. Are you getting my point? Those during selections, your expert discussion is also required. You can't select it randomly. You have just computer have given and you have selected it. No, you have to be discuss it with the experts in the field as well, and you have to go through the clinical data as well. You have to be go through clinical trials websites as well, to check whether there is any drugs who is undergoing under the clinical trial for any other disease. That doesn't matter, but any other disease, as I said, one target can play a role in the development of various disease. So if for example someone is doing a trial on particular targets on xyz disease, and by targeting that target, you came to know that no, no, this trial has been suspended, or this trial has been terminated due to this problems. So but you have to check the problem, because sometimes trials are suspended due to commercial reason or other reason as well. It's not the only the one reasons. Got it? Okay, so specific mechanism cannot be predicted. Yes, got it. Okay, next question is how can we predict particular pathway from the multiple pathway? Yeah, that as I said, of this, when you are getting the multiple target, you have to be understands the role of the pathways in the biological process. As I said, for some disease, for cancer, PI3K is good target, but for fatty liver, PI3K is not a good target. Whereas these both proteins, PI3K kinases, are playing a role in the development of fatty liver as well as the cancer as well, right? So depending upon what kind of disease you are targeting, that's why when we are talking about the networks, first you understands about the normal physiological process, create the normal, understand the normal intracellular signaling pathways and how they are altering in that particular disease, and then you have to select the targets for that particular disease. So by the computer, if you are getting multiple target, then you have to pick up which targets is playing a important role. Got it? To start with, next question is phytochemical can be extracted from the impact databases as well as for the in plant. Yes, means many researcher who are working in the natural compounds, they are pulling out these data from these databases, means structure of these phytoconstituents, and they are doing the screening. They are using the network and then doing the docking and other studies. Kindly repeat regarding adverse drug reaction. That I already repeated. In with respect to adverse drug reactions, you can identify the novel signals and then you can apply the possible mechanism. So there is a signals. There are two methods, frequency based methods and Bayesian methods. by which you can identify the noble signal of the drug, and then you can couple it with the network pharmacological techniques. Next question: if the source is animal, yeah, this is the questions I think that time I am reading, but not. If the source is animal, like hemolymph or mucus from snail, the identified compounds are mostly protein, can be used as ligand for the network pharmacology for human disease. Yes, why not? There is a protein target as well. There is a metabolite network as well. You can do it. See, network means there are nodes. Means node can be protein, node can be your drugs, node can be, means node can be anything. It can be metabolite, it can be proteins, it can be anything. So if it's a protein, we call it protein. If it's metabolite, we call it metabolite network. So there is no issue with that. Who is this? Momita Ray, 1987. Yes. Momita Ray, is it clear? So you can, there is no issue. Someone asked about PV signal identified by your lab, which database used and how. Is a FAIRS database is the public database, and we got the access from the IPC as well. VGBase, we have ADR monitoring center, so VGBase, ADRMS, UDRA vigilance of European Union. So all these datas we have pulled out along with the EHR, and then we have identified the signals. And you can go through our publication as well. We have done the publications by using this one, and I have authored edited book on signal analysis in pharmacovigilance as well, and separate book on network pharmacology. So in these books also we have written chapters with case studies how you can apply network pharmacological techniques in the signals. Got it? Next is, yes, audible. Sometime approved drug prediction target not providing the specific disease for which it is approved, and then how should we start the network study? Sometime approved drug prediction target not providing the specific disease for which it, you are talking about Sobit, you are talking about off label use of the medicines, you are talking about that. Like metformin is not approved for OCD, sorry, PCOD, but we are using it for PCOD as a off label drug. So there are many drugs which are being used as a off label, so you are talking about that. uh, yes, if you want to identify the possible mechanism of that drug in that particular conditions, you can do it. Yes, you can do it. There is no issue. Yeah, there are many off-label uses of the drugs which are not written on your SMPC or PI or CCDS, right? But physicians are using it, and you know that it's working. So you can identify the possible mechanism by coupling with the network techniques. Any other questions anyone has? You can put in the chat box. Now going ahead. Oh, it's 120. And my screen is visible. Can anyone confirm it? Okay. If anyone has any questions, start putting in the chat box. I will try to take your questions, each and every queries, right? So I hope till now you get an ideas where what is network techniques and where you can apply. And as I am saying, why not India? I always feel that because if you just see how much work across the globe Chinese researchers are doing on the network. Chinese are very fast, you all knows, and you will see in with respect to scientific evidences for traditional Chinese medicines, they have generated lot of data for their medicines. So that's why I always question is why not India. Yes, of course Indian researcher is also using, but not at that much that Chinese are doing. If you compare Chinese researchers using network techniques and Indian researcher, you will find there are huge differences there. So that's why I request every one of you use these techniques for generating the evidence for the herbal medicine as well. Of course, even though if you are not a pharmacognosy people, but you can use it for the herbal molecules, pharmacologies, other disciplines. Persons are also using the same. There is no issue. So before getting into the basic concepts of the network, as I said, every request every one of you, please read about important signaling pathways with respect to your disease, on which disease you would like to work. And this is a major mistake which I found everyone of us is doing. We are not focusing on the basic concept. There is no alternative for the basic. If you don't understand about intercellular signaling pathways which are involved in that particular disease, how you will understand the results of the network pharmacology? Don't believe on the computers. As I said, many times we have to exclude some targets because we know that computer is giving results based on these calculations. And according to clinical point of view, according to my knowledge, this is not the important, and you have a strong justification for it. So you have to exclude it. You can't do these techniques blindly, right? So that's why I always say, please read about your signaling pathways, whatever the disease you are focusing. But there are some signaling pathways which are playing role not only in the development of a single disease but multiple diseases, and these pathways every researcher should know about these. You can't say that you don't know about the apoptotic signaling pathway, because apoptosis is something which is involved in each and every cells, right? So apoptotic signaling pathway, every one of you have to know that how apoptotic signaling pathways, there is a intrinsic signaling pathway, extrinsic signaling pathway, how intrinsic signaling pathways are initiated, what is oxidative stress, how oxidative stress in mitochondria result in oxidative stress signaling pathways of the apoptosis, what are the caspase dependent, caspase independent, what are the proteins which are involved, how they are activated, how they are phosphorylated, dephosphorylated, and how they act on the DNA and doing the DNA fragmentations, which is a hallmark of the apoptosis. So you should understand about apoptotic signaling pathway or cell death pathways. Not only the intrinsic, these are the on the screen, these are the intrinsic signaling pathways of the apoptosis, which can be induced by DLM, is one of the pesticide which are widely used in the agriculture. How it can results in the apoptosis, that is explained in these figures. But if any one of you is working on cancer or in any disease, so you should know about these. You all knows oxidative stress and inflammation is something which is involved in each and every disease, right? So inflammations signalings also, you should, it should be clear in your minds what vascular events are started, what are the cellular events which are happening during the inflammations, how these inflammatory cytokines results in the intracellular signaling processes, all these things, right? So here is another one, is it can directly act on DNAs, and P53 signaling pathways is also can be bad and backs. These are the apoptotic proteins. There is a BCL2 which is anti-apoptotic proteins. So when there is a imbalance, BCL2 level is decreased and bad backs is increased, they are going to activate various proteins further, and then it act on the activate the caspase 3 and then do the DNA fragmentation. Again, I am just telling you quickly, maybe many of you knows about it, but again, as I said, these signaling pathways, you should, it should be clear in your mind. Like when we talk about phospholipases are activated by various external factors, and when phospholipases are activated, it do the degradation of phospholipid, and phospholipid is a major component of your cell membrane. Then your two products are formed, IP3 DAG is going to activate protein kinases. IP3 has a receptor on smooth endoplasmic reticulum, and smooth endoplasmic reticulum is the storehouse of calcium ions. More calcium is released, so how calcium is interacting, how it results in the DNA fragmentation, all these things should be clear in your mind before going to the network techniques. And of course, there is another things which is involved in each and every disease, the immune system. You all know immune system is everywhere, right? Wherever in the body you talk about, immune system is there. So you should know about immune system signaling pathway. If you don't know about immune signaling pathways, and when you create the network, you will get the target with respect to immune system, you can't explain to the expert in a better way, because you don't know the basis, you don't know what is the what are the targets which are involved in the immune systems in normal physiological process, and what happens in the pathological process in that particular disease, whether it's going to be activate, deactivate, or so what modulations, what happening with respect to immune targets. So Then for that one you should have a good understandings about the innate immune systems, adaptive immune systems, how they cross talk with each others. I hope you know about MHC, major histocompatibility complexes, and so on. I hope I am audible. You can post your questions in comment box. I will take it after the completion of this. So what I mean to say, read about these immune system signaling pathways. What are the PRR, pattern recognition receptors? When we talk about innate immunity, PRR, TLR is one of the example. You might heard about toll like receptors, nod like receptor, NOD, right? These are the PRR which recognize a particular pattern of the external substances or pathogenesis, and that pathogenesis contain the PAMP, pathogen associated molecular pattern. How the sensing is done, and after sensing, what intracellular signaling pathways are going to activate. It's going to activate MAP kinase, NF kappa B, then it act on the nucleus where it do the transcription translation process. There is a release of inflammatory cytokines, which inflammatory cytokines will release based on the nature of the pathogens which enters in the body. If it's a viral component, then there is a interferons. If there is a bacterial, then interleukin. Then you should understand which interleukin. Interleukin is also many types: IL 1, 2, 3, 4, 5, 6. There are so many, right? So which one is going to be, which one is anti-inflammatory, which one is inflammatory? So all these pathways, and how when your innate immune system is not able to handle that pathogenic substances by these innate immune signaling pathways, how it will be hand over to the adaptive part. So adaptive presentation is done by the APC, antigen presenting cells, and there is a role of MSC. If it cached by the MSC2, it will be presented to the helper T cells. If it cached by the MSC1, it will be present to the cytotoxic T cells. And based on that, it's going to be interleukin, and particularly interleukin 2 will act on the same T cells, that will be called autocrine signaling, and do the productions of lot of T cells, that will be called clonal expansions. Then there is a release of cytokines. Simultaneously, these cytokines will activate your B cells into the plasma cells, which will produce the antibodies. So, I am just quickly, I am just saying all these things to you, but you should understand about in detail, step by step, these signaling pathways. Again, I am repeating, that's very important, and that is a mistake most of the researchers who are working in the network is doing that part. Because without these understandings, everything is useless. There is no alternative for the basic. If you don't have a strong command on intracellular signaling pathways which are involved in that particular disease, what you will get from the computers? Are you getting my point? So these all are the pathways which we have studied multiple times. You should have a notes on it, and every times you have to search on this pathway. You have to search for the clinical trial on which targets with respect to immune system clinical trials are ongoing, what results are being coming, whether these are good target, druggable target or not. All these things you have to study it in details, right? When we talk about these particularly when we talk about enzyme linked kinase receptor, this is about the immune system. You can see that NF kappa B is there with respect to inflammations, but the inflammation is started when pathogen is entered. Then there is a different TLR, toll like receptor, TLR 3, 4, and there is TLR is particular TLR is sensing particular part, some sensing bacterial, some sensing lipopolysaccharide, some sensing viral components. So you should know which particular TLR is sensing, and after sensing, which pathway is going to activate, whether it is MYD88 dependent or independent, and whether it is NF kappa B or MAP kinases, and further whether it's which genes it's going to activate, and by the activation of that particular genes, which particular cytokine is going to produce, and after production of cytokines, what it's going to do. I hope it's not too much. So these are the presentations, how these are presenting to each others. There are dendritic cells, there is MSC1. Co-stimulatory signal is also required. So sometime in network you will get like these molecules CD40, CD20, 80, 86, and if you don't know about what is their role in normal physiological process, what is the purpose of network, right? If you don't understand normal physiology in a better way, then pathophysiology, then network, then pharmacology. it's a chain, without that you can't survive in this field, because you will face a lot of questions. If you are presenting somewhere in international conference, they will ask you a lot of questions. They will ask you how this target is important, this target already drug is banned, this that this happen. How you will say it's a druggable target for this? You should have an answer for all these questions. As I said, one target can be good for other disease but not good for another disease. You can't say this is a good target for all type of disease. No, it depends upon the disease as well, it depends upon disease progression as well. So unless and until if you don't understand that, you can't identify the targets, because identification of target is a huge task. I am talking about novel target. By doing all these analysis, you have to identify some new targets which are not explored yet. If you are doing these all exercises, if you come up with a already known targets which already everyone knows it, then they will ask you what is the purpose of this exercise. This already we know, this molecule is going to produce this effect by this mechanism. It's going to reduce the inflammation, we know it, and we have a scientific evidence by reducing the level of interleukin 2 or this one, this molecule is working. So why you have done this exercise? What is the purpose to do this exercise? Are you getting my point? So JAK-STAT is also important pathways. So read about these pathways. JAK-STAT, genus kinases. STAT is signal transducer activator of transcriptions. There are a lot of molecules under JAK-STAT pathways which are under clinical trials, and you have to understand which molecules are under clinical trial, which particular STAT molecules, and what is their advantage, what is their limitation, whether this is a good target for my disease or not. So stat 1, stat 2, stat 3 genus, and you have understands about the normal configurations of enzyme linked kinase receptors. In inactive state it's a monomer. When ligand is binding in the ligand binding domain, it's converting into the dimer form, then it's going to activate the kinases, then it do the phosphorylation of tyrosine molecules, create the active site for the stat. When stat is attached with that, it act on the nucleus and do the transcription translation process. Got it? I hope I am audible. I hope I am audible. Any question? So what I mean to say is I am not going to explain it in detail. If you want, you can check video on this YouTube as well. We have created separate video on JAK-STAT, separate video on enzyme linked kinase receptors, separate videos on other things as well. So there is a PI3K AKT is also another one, sir, which is nowadays many of you might heard about PI3 AKT, phosphoinositol 3 kinases. AKT is also called phosphokinase B. So sir, these pathways is also playing a role. How these are interacting, how these are phosphorylated, how dephosphorylated. Then there is a mTOR. We are talking about mTOR, mammalian target of rapamycin. We have a drug on mTOR, rapamycin is a mTOR inhibitor. So you have to understand what is mTOR, and mTOR also is of two types, mTOR C1 and C2. Which one is playing a role, which one is protective, which one is harmful, and how these are creating the problems. So that also you have to be checked. These are the mTOR signalings. As I said, there are a lot of molecules. PI3K inhibitors are also under clinical trial in discovery phase. AKT inhibitors are also there. There are dual inhibitors. There is a STAT inhibitors, mTOR inhibitors. But again I am repeating, it doesn't means that that particular target is important for your disease. It depends upon your disease pathogenesis, whether that target is important or not, and if you are blocking that, whether it's going to be produce the effect or not. Okay, so these are the discovery phases with respect to the signaling. So I request every one of you, whenever you are applying these techniques, correlate it with the clinical data as well. Otherwise, if you are not correlating it with the clinical data, you will always face the problems, and you will not able to defend the questions by the experts, right? So now starting with the simple network. Meantime, if there is any questions, any questions. User tech. Subham is saying, are you member of SRP at IPC? Soon this question is not relevant to network, right? Sobit is saying sometime approved drug prediction target not providing the specific disease for which it is approved and how we start the network. That already we have discussed. Next question is, sir, how we can select a new drug for any particular disease for repurposing? User tech, we can select a new drug by understanding the interactions of that drugs with other targets, right? So for that one, if you want to identify a new drug repurposing candidate, you have to be understands about interactions of that drugs with the other targets, and then you have to studies what are the role of these targets in that particular disease, and then you also understand the clinical data as well. I want to design, user tech is saying, I want to design my work on repurposing in neuroprotection. For that, how can we select a new drug? As I said, if you want to select a repurposing molecule for a neuroprotection, so first you have to understand about which disease you are targeting, because neuroprotection is a wider terms, so you have to be, select the disease in which particular disease you want to identify a drug repurposing molecules for the neuroprotections. Then you have to be understands about that one particular the repurposing. First you have to understand the pathogenesis of that particular disease, and then you can select the repurposing candidate by analyzing the clinical data as well. Clinical data's analysis is very important with respect to drug repurposing. Why? Because drug is already in the market, so you knows how drug is behaving in the large number of populations. So by analyzing the clinical data, it's little bit easy to identify the drug repurposing molecules for alternate indications. In that one, you have to be do the analysis by which you can get to know that if this particular medicine is giving to particular type of patients with comorbid conditions, so what is the effect on the endpoint which is related to comorbid conditions, whether it is increase decrease, so you can get a signal from there, okay, it could be effective in that particular disease as well. So with saying, yes sir, yes sir, I have tubulin ligand. Momitre 1987 is saying, I have tubulin ligand from snail and human p53 as a target for anti-cancer. Okay, in protein interactions, human proteins in same network is not coming as ligand target interactions. I have a tubulin ligand from this snail and human p53. Which tool you are using for the protein interactions? So for the these particularly, if you have a tubulin ligand snail, then you have to go for the you have a target as well, you have to go for the protein docking. Why you are going to the network part? You go with the protein docking, where you can check the interactions of, you have a ligand, you have a proteins, your ligand, both are the proteins, so you can go with the protein interactions. And to check the protein interaction, clust pro is the one of the online tool by which you can do it. Avdesh is saying yes sir. Rani Naidu is saying yes, audible. Yes, any questions? I hope I have taken all the queries which are there in the chat box. Still there is any questions, please put in the chat box. I'm checking on the mobile as well. There is any questions, or we can go ahead. So talking, starting with the simple network, I hope I am audible. Rani Naidu, I am audible. Avdesh, Momitre. Momitre, as I said, you go, if you have a two proteins, so you go with the protein docking rather than for the network, because you have defined your proteins, right? Clinical data, you have to be analyzed from the CTRI clinicaltrial.gov, and then you have to be check their efficacy endpoints. And sometimes you all know, sometimes the adverse drug reactions of the one drug, can also be useful for the other disease. And classical example is minoxidil, right? Minoxidil trial, it came to know that it result in the hair growth. Now minoxidil is in the market for the hair growth. Like that, there is a sildenafil, the repurposing, where so adverse drug reactions of that particular drug is being used for the therapeutic indications, right? User tech, user T6SH4WK5H, बड़ा difficult नाम रखा है. Anything else? Okay, or we can move ahead. Okay, great. So simple network. So as I said, there are a circles. That circle we call node or vertices, and that circle can be gene, can be protein, can be metabolite, and so on, right? And in every networks, whatever the network you will see in the research articles, you will find there is a circles, and these are circles are connected with the lines. So circle we call nodes. Somewhere someone is saying it vertices as well. So node or vertices are the same thing, and that can be depend upon what kind of network you are dealing with. If you are dealing with gene network, so it can be gene. If it's a protein, so it can be protein network. It can be metabolite network. So line connecting these lines which are connecting these nodes, we called edges or links, right? And that tell you about the relationship, how these nodes are connecting with each others. So this is just a simple network, and it's based on the graph theory. As we have discussed about these network techniques are based on the graph theory, and graph is equal to combination of vertices or edges. So vertices is nodes, and edges are telling the how these vertices are connecting with each others, or you can call it nodes or links. So graph is a combination of vertices and edges or nodes or lines. Are you getting my point? So this is just a simple network. So if you are seeing any simple network, you will find there are circles, and these circles are connecting with each other. So circles ko we called it nodes or vertices, or the line which are connecting we call it lines or links or edges. So we represented with the N or L or vertices V and E, right? So when we talk about graph theory, so this is the graph theory, on which network depends upon. So graph is the 18th century by the Swiss mathematician Euler. So there is a famous Königsberg bridge problem. So there is a Königsberg city is there, which is connected by the seven bridges. One, two, these red are the bridges. One, two, three, four, five, six, seven. This is connected with the seven bridges. So this problem is means not problems means this is a solution is provided by the Euler mathematicians in the form of graphs. So this is the origin of the graph theory actually. So the as I said, the German city of Königsberg is situated at the Preguilia River, and geographical layout is composed of four main bodies of the land connected by total seven bridges, right? So the question posed to the Euler is the mathematicians: was it possible to take a walk through the town in such a way as to cross over every bridge once and only once? This is known as a Euler walk. So is it possible? So so all the residents who are there in the city, they know that it's not possible, but they don't have any evidence for it. So Euler mathematicians, with the help of graphs, they have provided the evidence for it, and that is the origin of graph theory. Got it? So graph theory is ultimately, what is graph theory? If someone asks you what is a graph theory, you are talking about with respect to network graph theory. What is graph theory? So graph theory is the study of the relationship with each others, right? So network is based on that. And when we talk about different types of graphs, so graph can be directed, graph can be undirected, right? So undirected graph, so you can see that these circles are the nodes, and the lines which are connecting these circles we call links or edges. So if we talk about undirected graph, so a, I, j, these are the nodes. If I is the one node, j is the another node, so this a, I, j is equal to j, a, j, I. What does it mean? It means that you can go from 1 to 3 or 3 to 1 in both the ways. So there is no directions, right? It can go from 1 to 3 or 3 to 1. It goes both the ways. So it's a bidirectional. So it's a undirected graph, right? Where there is no directions. Are you getting my point? So such kind of network where no direction is given, that we call undirected one. And undirected one, we are mainly when we are creating the protein-protein interaction network. These are mostly the undirected one, right? Phosphorylations of this protein to dephosphorylations, and goes vice versa as well. So this kind of graphs we call undirected graph. Got it? And directed one is, you can see in the directed one, there is a direction is given. Direction from this node to this node, this node to this node, this node to this node. So direction, when direction is given between the nodes, such kind of graphs we call directed graph. So in directed graph, Aij, I is the one node, J is the another node. Whether they are equal? No. This one you can go from 1 to 3, but can you go back from 3 to 1? No, you can't go back, right? So such kind of graphs we call directed graph, where direction is given. So that particular, when we talk about pharmacologies or biology, we call it gene regulation networks. Then there is another kind of graph is particularly weighted and unweighted graph. So you can see on the screen, weighted graph means you are giving the weightage between the two nodes. I have given the weightage from A to C, 10 weightage. Here A to D I have given 2 weightage. C to B I have given 1 weightage. A to B we have given 4 weightage. So weightage can be provided based on various parameters. One of the parameters when we talk about text mining approaches is the frequency of that particular terms in the document. If frequency is more, we are giving more weightage. If for example association between this node is much more stronger, you are giving more weightage to that. So such kind of graphs where value is written between the two lines, such kind of networks we called weighted graph. And where no weightage is given, we called it unweighted graph. We are not giving any weightage from one node to another node, right? Then there is a another type of graphs that we called acyclic or cyclic. So you can see that acyclic and cyclic graph can be of two types, directed or undirected. So you can see that in acyclic, even though it looks like cycle, but I am saying it acyclic. Why? Can anyone answer it? This graph is acyclic, but it looks like cycle, right? Cycle means it's completed, means it looks like circles. So it looks like cycle, but even though I am saying that this network first one is acyclic. Why? Can anyone answer this question? And of course it's a directed, this is a undirected, because here is direction is given, here no direction is given, that's why it's a undirected. Here direction is given, so it's a directed. But still I am saying it's a acyclic. Can anyone answer in the chat box why this is acyclic? Yes. Anyone can put in the chat box. I'm waiting for your answer. My question is, first one is it looks like cycle, but why I am putting it in the category of acyclic? Vijay is saying bidirectional. Isa is saying lines from A goes to B. But I think Isa, can you put it again? Can you put your answer again? Oh yeah, now it's visual. Lines from A goes to B and as well as C. A to B and then C. That's why it's acyclic, you are saying? Anyone who cycle is not formed? Yeah, Subham is saying right. Cycle is not formed, right? Start with any node. If you are starting from C, you can go from C to D. But can you go from C to B? No. Direction from C to D is not there. Yeah, user tech is also right. So cycle is not formed. That's why it's a cycle. You start with any node. You start with B. You can go from B to D. Whether you come back to B, no, right? You start with C. You can go from C to D. Whether you come back to C, no, because from D there is no direction to B, right? That's why it's, even though it looks like cyclic, but it's a acyclic. So when you are analyzing the graph, please check whether it is a cyclic or acyclic. So now you can see that it's a cycle. You start with any node. If I am starting with D, so I can go from D to C, C to A to B, and B to D back. So it's making the cycle, right? If I am starting with A, I can go from A to B, B to D, D to C, C to A. If I am starting with B, I can go from B to D, D to C, C to A, and again B. So cycle is formed. And of course there is direction is given, we call it directed. Here direction is not given, we call it undirected. So someone is saying, Vijay is saying bidirectional. Bidirectional is this one. Undirected graphs are bidirectional. Bidirectional means there is no directions, it can go from D to B and B to D as well. So Vijay, that is a bidirectional. Undirected graphs are bidirectional, but directed graph is not bidirectional. Vijay Pujita 255. Are you getting my point? So undirected ones are the direct bidirectional graphs, right? So next types of graphs are, it can be sparse or dense. So so sparse means when there is a few connections, that kind of graphs we called sparse, and when there are a lot of connection, we call dense graph. So when we are dealing with network, biological or pharmacology or disease, so these we are so dealing with the dense graph rather than sparse graph. So if someone asks you difference between sparse and dense, so we are dealing with the dense graph rather than sparse graph. These are the different types of network we already discussed. It can be proteins, it can be metabolite, it can be gene regulation, it can be cell signaling, it can be protein. It depends upon. And these circles in every network represent the nodes, or we call vertices, and these are the links or the edges, right? So don't forget that part. The another types of networks, when we talk about small world network, scale free network, random network, modular network, or bipartite network. So what are these network? A small world network means if distance between the two nodes is less than 7 steps, so we consider a small world network. So small world network, I always give example. If you are going to some conferences and meeting with some individual, oftenly say one word, uh, see, this is a small world by which everyone is somewhere connecting with each others, directly or indirectly. So that is small world networks where distance is less. Scale free network, you can see the differences between a scale free and small world, where there is a connection is not there. Random network, randomly they are connected. Modular network, where you will find small modules which are connecting with each others. So you have to be understands what kind of networks is created with respect to your disease pathogenesis, whether it's a modular network, and whether this module is important. Sometimes the connections, node which is connecting these module, that is more important to target rather than these modules. So it depends. So this is the bipartite is particularly we are dealing with the when we are talking about the network pharmacology, because we have a drug as well as the disease. So that is a bipartite network, is particularly with respect to network pharmacology, because network pharmacology we have a drugs and the disease, right? Yeah, Momtire is saying direction is not continuous in the first one. Yes, you are right. So types of network we already discussed. There can be gene regulation network, protein network, signal transduction network, and metabolite network. So now next is your hands-on sessions. Now steps. So are you with laptop? How many of you have a laptops with you or computers? No. Okay. So steps. What are the steps? If any one of you now you have a enough basic idea about network, Now you can ask me, sir, now I want to apply these network techniques. So what steps I have to take? So steps which you have to take is take is particularly you have to identify the targets which are involved in the disease. That is the first step. Or you can identify the target which the compound can hit. So to identify these drugs, identify the targets which are involved in disease, you have to be pull the data from the different databases. So you can see that on your screen. This is a which you have to do, where you have to be pull out the targets which are involved in particular disease. So to do that, yeah, I hope my voice is clear. So you have target platform: DigiNet, GeneCard, OMIM, HPO, Malacard, Orphanet, GWS Catalog, Hughes Navigator, CleanGen. So these all are the freely available server from where you can pull out the targets which are involved in that particular disease in which you are working. But now, important is, depending on your disease, you have to be select the databases. There are some databases like which are for the orphan disease. So if you are working on the disease, so you can't miss the orphanet data, right? This is the first steps where you are, as I told you, first you have to be understand the duplicate, remove the duplicate, and make the one comprehensive file. And text we also discuss, but just overview about the steps. These are the first steps which you have to do. My screen is not visible. I'm going to share screen again. Identification of target of compounds, so that your compound can hit. So that one, Swiss target predictor, C database, stage drug mining. There are others as well. So this is the second step, and these are the two steps which are the foundations for the network. So you have to be give lot of time for these two steps and lot of verification as well. you know so I will explain you what are the cutoff values and how to select it. And it's just an overview about the steps which you have to do. And then you can identify the common target by Vani or by advanced Excel as well. Then you check the interactions among the common targets by the string, biograde, deep stitch, inact is there. And then you can create and analyze the graph using the network graph I Python. So I request every one of you use these or R package, because the beauty of these two is it's a freely available, and you can do lot of analysis by that. Uh, Much more analysis, but based on the commands you can calculate the parameters, right? So network x, if you are using the Python, you can use it by the network x. igraph is the module of R package. Maybe many of you are using R package for analysis as well, or other analysis like disproportionality analysis, which adverse drug reactions, right? So that also you can. And then you can visualize or make beautiful networks by using the cytoscape. And you can do the pathways or enrichment analysis by the SR. After that you can go with the docking studies and the molecular dynamics. Then it will become a complete set, right? So first step is target involved in disease, but I think time is already over. So now I hope you got an overview about what are the steps you have to be take. So tomorrow I will try to be in the good internet connection, so that we will start these steps. So you come up with your disease, you come up with your research questions. So I will try to explain you how you can do it step by step. So your means be with your laptop or internet connection, so that whatever the steps I will show you, you can do by yourself, so it will give you the confidence much more, right? Meantime, you have any questions, any things, you can put in the chat box. Anyone has steps, you can save the screenshot as well. And also, so apart from these databases, if you came to know that, you go to the literature, you came to know that there is another web server as well, which is not listed in this list. Include that. you know you will feel that why I am saying same things you have to do in all the databases. Reason is because you are using the freely available databases. The reason is most of the time they are not updated time to time, so you don't want to miss any target, because these are the foundation for any network. So I always say, give much more time to these two steps, because after that whatever you will do, that are based on these two steps only. So don't do mistake in these two steps. Otherwise what will happen, when you are not there in the network, what is the reason for it? So how I will believe on your results, how I will believe on your analysis? So so you have to be very careful when you are selecting the target. That's why you can see at the end I have mentioned text mining approaches also we have to do, right? So that also I will explain you what are the text mining approaches and how to do that. Any questions? Any question anyone has? You can put in the chat box if someone has any questions, anything. Anyone want to say anything? How many of you have already done the network? Is there anyone who has already done the network, sir, for any you have already done the network project, or you all are beginners? Yes. Yeah, Isagar is saying please show the slide, identify. Okay, Isa, you can take the screenshot tomorrow as well. So we will do these steps, sir, in step by step in detail as well. Tanisha is saying no, sir. Okay, you are new to this, Tanisha. Okay, so meantime, as I said, read about intracellular signaling pathways, and some of the pathways, as I said, playing a role in the development of each and every disease. So there are many videos which are available, which explain about JAK-STAT pathways, which explain about so mTOR pathways, which explain about enzyme link. Everything is there in the this one on YouTube as well. You can go through it and understands the pathway, then you can do the analysis in a better way. Okay, if there is no questions. New, Subham is saying new. Okay, you are new to that. Okay, okay. Yes sir, beginners. Okay, someone has done. Please show the slide identification, beginners. Jan is saying I have done on Gamia Pinata. Okay, great. So I think if someone have already done, so you knows how comprehensive this exercise is. It takes a lot of time, then it will make very comprehensive, and you get a lot of confidence. Wherever you are telling about your research, you will be very confident, right? You have done each and every steps in a standardized way, and you have take all the care when you have select the target, when you have prepare the files. And again I am saying, correlate your findings with clinically or post marketing data or already available literature as well. Then only you can you can connect with that. Otherwise sometime what happens, for example, you have done this analysis, you have identified some targets, and researcher or expert has asked the question, this target is not good for this particular disease for which you have identified. And for example, you have done experimental work as well So it become lot of it create lot of tension to you, right? Whatever work you have done, you have select wrong target, and already you have planned your best plot, or you have completed that one as well. And expert said, no, 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 for this disease this is not a good target. How you have picked up this target? That's why I'm saying, once you done these all analysis, everything, you have to be discuss it with the experts. You can't select the target randomly, because after that, if you are going for the validation of the targets, so it's a expensive process, right? So selection of target is very important. Yeah. Yeah, Janice is saying how to go for the geo analysis. Geo analysis we will do tomorrow, because first I can ask all of you to prepare the files for any targets which are involved in disease. Then you can create the disease network, and tomorrow we will create as well as do the analysis of the network. So I think if you are comfortable with the iGraph, you can do with the R package. And how to install, yeah, one thing you all can do is, before tomorrow sessions, install iGraph, install R or Python in your systems. And how to install it, how to install particular module, and how to use it, YouTube video is available on the playlist of the network. In this particular playlist, you will find that there is a videos where we have explained about how to install iGraph if you are comfortable with the R package, and how to install network X module of the Python in your laptop. So install it, then only you can create and analyze the network. And in videos also, it's explained about so how to create and analyze as well. There is a separate video on that. One of our scholars have explained it. Okay, Israel Lokoro is saying beginners. Okay, great. Yeah, beginner is also good. So I hope every one of you got an ideas what actually these techniques is. Now you can think of where you can use in your research, right? Any techniques for your research. It may be, because after this session, maybe it's not important for you. Maybe it's not according to your research. It's not applicable for you. So it's not like that. You have to be, you knows these techniques, so you have to be applying it. You should have a strong reason for it, why you are applying it. By applying, what you are going to get out of it. That should be clear in your mind, right? Anything else? Anyone, you want anything else? And of course, with respect to network, how many of you knows about network meta analysis? Anyone who knows about network meta analysis? That is also, somewhat similar. Means we are talking about there also nodes, or but of course there is a different set of data, different modules are there for the network meta analysis. But very interesting, where you can compare one treatment with another, which is not compared in the clinical trials here, but there is a common comparator is there. So for that one also, there is a means we have done one workshop on network meta analysis as well. You can get that information as well. Anything else, or I can close the session? So sorry for internet. Connection is not good today, but it's always happen. Uh, for such kind of sessions, network is not supporting. But of course, tomorrow I will try to be in a good connections. And I request every one of you also come up with, when sit with the, not come up, sit with your systems or laptop, right? Okay, then thank you everyone. Any questions? Let me see at last. Any question is there? Beginners. Okay. Okay, then thank you everyone for your time. Have a great day. Enjoy the learning for the network. Apply these techniques where you want, right? If there is, and I also explained you how to couple it with the experimental techniques, right? And means how you can overcome the problem of the experimental validations. As I already explained, means many times we don't have that facility where you can do much more detailed target analysis or so thing. But another thing is, of course, you couple it with after. I think molecular docking is something which everyone of you know, right? By using Autodock, or there are many Swiss Docker. There are many tools by which you can do the molecular docking. Of course, for molecular dynamics, you should have a good computational systems. But few molecule you can do for the MD, then it will become a complete process. Okay, so thank you everyone. Thank you. Have a great day. Thank you.